Hello again, everyone. Welcome to Q&A with A&V. I'm Vincent Racaniello, and with me from Columbia University, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
I'm well. How's your
They're
black
still
essays?
talking. They're good. They're brewing. They're still talking about the ice cream. So this is the closest because I do like pistachio, but it's not actually my favorite.
What is your favorite?
So I actually like cinnamon. No, I'm serious.
Yeah, so after Bobble said if pistachio, but you like cinnamon ice cream?
Yeah, homemade
Yeah,
cinnamon ice cream.
and Andrew had said also
Yeah,
pistachio.
yeah,
Okay.
yeah.
Um,
Now they're fighting over who said it first, but it's like it's cute. But I do like commercially available would be pistachio, but when I make it at home, it's cinnamon.
all right. Now we have a comment here that I think is funny.
Okay, which is
Those who think COVID-19 vaccine will modify their DNA should see it as an opportunity. Sign posted on at Ross Story
that is good.
as an opportunity to improve themselves, right?
Apparently,
So Eileen bought an at-home test kit. So this is the Abbott something now. Or what is it called, Amy? Do you know the name?
I don't know the name.
Binax Now.
Sounds familiar.
Let's look at anyway. Abbott Binax. I thought that was the name of it. It's the one that that the government bought up. Well, Eileen, when you get it, if you take it, let us know your experience, okay? Like how it worked and what the results was and so forth. Hello from the UK. Welcome. We had people from Peru this afternoon in Tuiva, Amy. Oh,
That's cool.
Plinky thought it was butter pecan. That wouldn't be Amy, no.
No, that's my sister.
I don't
But
know
she
if this
also
is.
likes cherry vanilla, which is just yucky.
I don't know if this is fair, but Kathleen wants to know which size shoe you wear.
Depends on who's cutting the shoes. So it varies between a 7 and a 7.5. And if it's really like a Christian Louboutin, it's an 8. I have small feet.
What are your favorite shoe brands? Louboutin, is that one of them?
He's okay. He's
Loubou-
very like Christian. I think the red heel is really great. I think that his signature is really great. Um, but I actually really like a lot. I have a lot of Manolos and Chanels and some Valentinos.
Which I never see because she doesn't wear them to the lab. So
No, I don't wear them to the lab, but I don't wear my good clothes to the lab either.
no.
No point.
Logically, doesn't an inactivated vaccine make the best due to more epitopes? Well,
No, logically, infectious attenuated vaccine makes the best, and inactivated is second best because of more epitopes. Yes.
yeah, I mean, it does have more epitopes, but I don't know if that makes it the best automatically. It depends. We have to check it, right? What do I do when we reach 100,000? We'll have a celebratory live stream to thank all of you. And I think that's great because the more subs you have, the more you get. And 100,000 is a good goal. And then we keep going. Then we go to 1 million.
From 100,000, you're going tenfold.
All right, let's go
What
to
happened
200,000.
I was going to say let's go in increments.
Okay, 200,000. But all right, so viruses don't actually have colors. Media strikes again. You know, I wrote a blog post years ago, but nobody reads my blog. It was called "What Color Are Viruses?" And I ought to put the link in. Virology blog, what color are viruses? And actually, it was a very clever post. What color is a virus? There it is. I'm going to paste it into the chat. And should you be interested, read it. I did this 8 years ago, 2009. long before the press thought of it. I'm way ahead of them all. And Amy's rolling her
That's
eyes because
12.
I'm ranting.
Yeah, and it's 12 years ago.
What do you
You're not
what do you
very good without the big button calculator now, are you?
Okay. We were on a call today, and our collaborator said Amy doesn't pull punches. Isn't that what she said?
I don't know. I wasn't. I don't remember.
Morris says, "What do you think is the motivation of fearmongers like Hazeltine and Osterholm?" Do you have any insight, Amy? Before I go,
Motivation? How am I going to know what motivates them? I'm not in their head. Um, so I'm not sure I would use the word motivation.
I
I
think
think they are just ignorant, and they have a platform and nobody corrects them, and it propagates like rabbits. But I don't know that I would use the word motivation.
I think they're uninformed to a certain extent. And I had an argument years ago with Osterholm, and I said he was wrong, and he just said, no, I'm right. So the people, they don't want to take any criticism whatsoever. It's not worth discussing. Let's cut to the chase. Are we all going to die?
No, we're
No.
going to be fine.
We'll be fine. Think of it this way. 1918, we had no interventions, right? It burned out in a couple of years. I mean, a lot of people died, unfortunately, but it
Burned out
stopped.
in 5 years.
5 years?
5 years. Yeah. It was a pandemic for 5 years. However, there was a whole big National Geographic article that talked about, how vaccination is game changing in the terms of it's now a young person disease. And they're not really wrong.
No, they're not wrong. Okay.
It's just unfortunate that I then read another article back to back with that, but in some other site that talks about how 20% of the elderly refuse to get vaccinated. I was like, that's a good plan. Good plan. Let me know how that works out for you.
Okay. Headlines today. India's double and triple mutant COVID variant. What's going on? The media doesn't know what's going on. There have been double and triple mutants all over the place. I mean, there are many mutations in these variants. What's the big deal? So there are a couple of spike changes in one. It's not an issue. What do you think, Amy?
I don't know of any variant that just has 3 mutations. And so first of all, mutation is not the right word, right? Because we're talking amino acids, so it's alteration. I don't know any of them that only have 3 alterations. I don't know that I'm unaware that they are novel and have not been seen before. I'm more aware of the fact that they are not appropriately dealing with the public health emergency.
Okay, here's our first J&J vaccine question. Do you think it's as bad as the news is reporting as far as blood clots?
Listen to Daniel on Saturday.
Last Saturday or coming up?
Both.
I mean, he seemed to be agreeing with stopping with the pause in the U.S., right?
Yeah, but I think he believes that there needs to be more data.
I also think that, didn't the EU, the European Union, say you may take it and understand that the benefits outweigh the risk, right?
Yes. But Daniels also said that, so the EU is not a good comparison because they don't have enough Pfizer and Moderna.
Yeah.
They actually don't have any, I think, or very little. And whereas here, it's like, 100,000 have, let's say 100 million have been given, and those are Moderna plus Pfizer, and then 1 million of the J&J. So it's 1%. It's not a big deal. He says we can overcome it.
Remember, you get clots from COVID also, right? You get 10 times more likely.
Right. You also get clots from the mRNA vaccines. It's just very quiet.
All right. Abraham says a study showed reduced vaccine efficacy in people with leukemia when measuring IgG. In people with no IgG, do you think they make at least a T cell response?
So my parent has lymphoma, and they are treating it by reducing B cells, right? So she has the reduced ability, or they have the reduced ability to make antibodies, and she's fine. She's been vaccinated twice. She's fine, and her T cells look great. So we're not concerned.
Any concerns of mosquito, tick, bed bugs, mites, lice transmission? I'm
Where?
not concerned. The virus doesn't reproduce in these animals.
Oh, it's about this virus?
Yeah,
Okay.
not other
Okay.
viruses.
I didn't understand what he was talking about, actually. But yes, it is not an arthropodic virus.
Someone just said, and we'll get to it, to be pedantic, everyone is going to die. And that's true, right? That's what you would say, Amy.
I do. I do say that.
Yeah,
a
we're all
lot.
going to die. When I come in some mornings, I tell Amy, I'm dying. And she said, we're all dying.
And then you try to tell me how you're dying faster than everybody else. And I just pull my glasses down and look over them and say, really? Then be quiet.
Robert says, should it be a concern that only 0.2% of the world's population have had the vaccine and 80% of countries don't have access?
Of course it should be a concern.
It's a huge concern. And you would think the mainstream media would emphasize that instead of double and triple mutants, which may not mean anything. But no, they want to emphasize we're all going to die. I should say faster.
Faster? Really? How are you dying faster?
Well, if you get serious COVID, you could die in a few weeks, right? As opposed to in 20 years. That's what I mean.
Oh, okay. Because I was thinking it's like when I used to go to the Kentucky Derby as an undergrad, and they would say it's the fastest 2 minutes ever. And I'd be like, it's 2 minutes. It's 2 minutes is 2 minutes. I don't understand how it's the fastest 2 minutes ever.
What is gain of function research, Amy?
Gain of function is where you weren't able to do that task, and now you're able to do that task.
Yeah, so you give a virus.
Variety. It comes in a variety of different forms, right? So when we talked about taking 2 dead polios and getting a polio out, that's a gain of function. But you really like only to think about it in terms of pathogenesis.
No. I mean, drug resistance could be gain of function, right? Temperature sensitivity or any new property you give to a virus is gain of function. That's all.
It's anything. It's not a virus. It's anything.
Oh, yeah,
whether or not you give it to a mouse or to a bacterium, it's anything.
But as you know, Amy,
across the board.
I think viruses. I dream viruses, Amy.
Okay, dream away. Let me know how it works out for you.
What color do you think those dreams are in?
Black and white.
Charlotte wants to know. I have to teach face-to-face with the college kids. I guess in the fall you're talking about? I will wear the mask. I've been immunized. I have nervous 36 students. Is it dangerous? Well, they should be immunized by the fall, right?
For sure. Many colleges, so Columbia, NYU just decided, Yale, Rutgers, Cornell. They've all, a couple of places outside the city whose names forget me, they've all decided that you cannot come back to campus if you're not vaccinated.
I think you're fine, Charlotte. Masked, vaccinated, no worries. Teach them good stuff. What do you teach, by the way? All right, Samantha, this is my favorite study break. Yay, from Chicago, which is a former Amy place.
It is. I used to live in the West Loop on Monroe.
Smash the button, the like button, everyone. That's just below the video. We'd love it if you could smash it, but don't break your computer.
Cute dog, though. Now that looks more like Gordy.
You think?
Well, Gordo is a schnauzer. That's not exactly a schnauzer, but it looks more like Gordo. Because Gordo also has a white chin.
What is the fate of defective particles? Could they play any role in persistence of detectable RNA long after recovery from COVID?
They're usually released and cleared. So
Yeah, I
I
think they're
don't know
cleared. Yeah.
that. I don't think that they're detectable RNA. The RNA is sitting in the cytoplasm. I wouldn't consider that a defective particle.
Yeah, I think they're cleared. I don't think they have a role in persistence. Arthur is having dreams of epitopes, endogenesis, and all sorts of weird stuff. Yay, we achieved our goal, Amy. We've got people dreaming about science.
Excellent.
Charlotte thanks Amy for the Calder exhibition at at MoMA.
Yeah, it's supposed to be great. It ends right before my birthday. Hopefully, and the museums actually are going up to 50% capacity on Monday.
What are they now?
25. But
According to Plinky, Hazeltine is an aggrandizer and Osterholm is misguided.
okay, we can go.
I
with
think
that.
Osterholm
like
likes publicity. I think Osterholm likes publicity, and he doesn't always check his facts. All right. Thank you, Post Polio, for your super chat. Quote of the day: association and correlation are being missold to the public by the ill-informed and ill-intentioned as fait accompli proof of causation. Yes, that's right. Association and correlation are not causation.
So this
right.
reminds me of when I was a graduate student, and we used to read these papers, and they would come to me, and my response 9/10 of the time was guilt by association. Gavel, please.
Trevor wants to know what's the deal with the variant in India. So the variant has 2 or 3 changes, spike changes that were in other variants, and at the same time they have. I think of the changes of concern, the ones either.
Yeah, there's other ones, but yeah.
Anyway, and at the same time, there's a big increase in cases. So of course, that's the association which we just talked about. The people are saying, this double, triple mutant must be causing the increase. So it's an association. It's not causation. Charlotte said,
go through this last month with somebody else.
If I had the time, I would make you a mobile.
Oh, thank you. Thank you.
Sean says, my friend thinks he can catch COVID from me because I was vaccinated last week. Should you find smarter friends? I think you should become friends with Amy, and then you get all your questions answered, right?
Yes. Yes, you should find smarter friends. I had to tell one of my very close friends that his partner might have to reapply. Then he said, he pointed out a few other things. So then we said, okay, we'll take it back.
Don't most proteins autofluoresce if you hit them with UV? You don't think so, right, Amy?
Not that I'm aware of.
No, that's why you need proteins like green fluorescent protein.
There is a background level of autofluorescence.
But it's low enough so that GFP will be much higher than that, yeah.
Yeah.
Why do you think there seems to be more anecdotal side effects from Moderna compared to Pfizer?
Probably what's in the lipid.
Yeah, but I don't think it's really a concern, is it?
No.
Okay, here we go. This is MK who said, to be actually somewhat pedantic, we're all going to die. Thank you. You're right.
We already addressed this.
I am due for my second dose next week. Severe reaction lasted 14 days. Any chance the next one won't be as bad? Usually the second one is worse than the first one. So, I'm sorry you had a 14-day reaction, but maybe it's done. You never know. I wish you luck. Take care of yourself.
Listen to Daniel. Somewhere he's probably talked about this.
Yeah, Daniel said 425 is the end of the period when the people who got the last doses of J&J will go through the time when they would get the clots, and then they'll decide what to do.
No, 425 is Sunday. Are you sure it's 425? Because the committee's meeting on Friday, which is 423.
Daniel said 425 also.
Oh, okay.
When is the latest I can send a question to Dr. Griffin? So we record on Thursday nights, Arthur. So you got to get it in tomorrow.
Tomorrow by noon.
Yeah, because I'm going to select them in the morning. Would I be nervous going back to back? Actually, I was going to teach a class in person last week. I had arranged to do it a while ago as a guest lecturer, and they told me, no, we're nervous. We don't want to get together. But I would have gone because I'm vaccinated, the other professor's vaccinated, and it's a small class spread out, and everyone would mask. So I thought that would be fine. But Amy, what is your opinion about mold on cheese? If you cut it off, is it safe?
Don't you need mold to make cheese?
Yeah, but then after in the fridge, it may accumulate mold that had nothing to do with the cheese making.
Sure,
I cut it off.
And, I was going to say, sure, cut it off. Plus, some of the rinds, but some of the rinds you eat. Like, don't you eat the brie rind, which is mold or something?
Yeah, some cheeses, definitely.
Some rinds you do eat the rind for. I mean, I don't eat a lot of cheese because I'm allergic to milk, but I believe that some cheeses you eat the rinds.
That's true. The brie rind is mold. So I cut the mold off cheese all the time. My wife says I'm going to get sick, and I never get sick, okay? So that's an N equals 1. But I think if you cut it off... I'm sure
there are millions of other people who do this, so there's not just an N equals 1, number one. And number two, I don't know. I think there's mold on goat cheese. It's the white ash stuff. Or actually, it's ash on goat cheese, and people eat it all the time.
What do we know about effectiveness of vaccines after one dose? AZ, Pfizer, Moderna, JJ. I'm going to let Amy answer this because I always get in trouble when I do, right?
I don't know. Do you get in trouble? Well, Pfizer is a one-dose vaccine. I mean, J&J, a one-dose vaccine.
Right.
There is some data that is suggestive that Pfizer and Moderna one dose is fine, and then you just delay the second dose for a couple of months because they went in thinking that they were going to give 2 doses, and so they made them very close together. So actually, when it's rising after 2 weeks of the second dose, you really don't know that that's because of the second dose or that's because the first dose wasn't finished rising when you took the second dose. So it's flawed. And AZ, I thought was 2 doses, but I could be wrong because it's also an adenovirus. Yeah, it is.
AZ's 2.
Yeah, and I don't know really that much about the AZ vaccine. So because it's not well described here. And
the AZ vaccine was studied. We did this on TWIV a while ago. They found after the first dose, they had some people who didn't get the second dose right away, and the immunity kept rising, and the protection was quite good. So that's part of the reason they delay the second dose in the UK as well. So, yeah, I think a long gap between the doses is not a problem. Everyone is saying that cheese, cut off the mold, and someone asked, is there cheese in the vaccine? It's good that you guys have a sense of humor.
Yes.
Given the effectiveness of N95, why was there never an effort to ramp up production? I don't know. I'm not a supply chain guy. Are you, Amy?
I am not a policy or economist, or I don't have anything to do with that. I'm not interested.
Okay. Let's see. I'm going to, we just have AstraZeneca. Should we advise young females not to take it? Well, I think you have to follow the advice of what the country is telling you. Now, if you're in Europe and they're saying do it at your own risk, what was the age range? 18 to 58 in the females, Amy?
Yeah, somewhere around there.
I think you need some kind of vaccine, right? That's the point, because not getting a vaccine can be worse.
Yes.
And that's the issue. Abraham says, we love you, but your mom seemed to seroconvert. My question is for those who don't. Do they at least make a T-cell response? I think it depends
Yes.
on
I'm
the
not
type
sure
of...
that my mother seroconverted.
Yeah,
I
you
don't
didn't
believe
say
that
that.
she did. But they do. A T cell response is independent. It comes first.
Fear-mongering scientists, their plan may be to scare people into behaving more responsible. At least here in Italy, I have this impression. Yeah, I've often thought of that after Babel. Yes, they could, like Peter Hood says, might just want to scare people, or even policymakers who should know better. They maybe want to scare them. That's a reasonable thing to say. Yeah.
I don't think it works out that well.
I think this is great delineated. Next headline, virologists discussing COVID say we're all going to die. That's right. They're going to take a line from this live stream. It's going to be headlines tomorrow, Amy.
Okay. Let me know what paper so I can subscribe.
Are the cells in which the vector viruses would grow naturally permissive or have they been modified? Well, if you're talking about adenovirus vectors, the cells would be permissive and susceptible to wild-type adenovirus, but this is E1 deleted, and so the cells are modified to produce the E1 protein so that you can grow up the vector. And that way the vector doesn't reproduce in people.
Mm-hmm.
If original antigenic sin applies to coronavirus, would an N protein-targeted vaccine give a second chance at a new effective immune response to those who haven't actually had COVID?
N protein does not produce neutralizing antibodies.
That's right. No, so it makes T cells, but that wouldn't be enough for a vaccine, I'm quite sure.
Not clear how it would work.
Ralph Barrick passaged SARS-CoV-2 in mice to be lethal. Do you think this path of evolution is likely to be followed in humans? No. Absolutely not.
He didn't exactly passage SARS-CoV-2.
What did he do?
He made a virus based off of informatics that replicated in mice using ACE2, and he passaged that virus.
That's correct. Big difference.
Yes.
The Amy Appreciation Club is here. Look at all those hearts, Amy.
It's cute. It's very
yes.
cute.
Okay, Charlotte, if you're teaching in the fall, you're good. You're all good. All your students are going to be vaccinated.
Everybody under 12.
My 11-year-old asks, can viruses fight each other? In a way, they can interfere with each other. Yeah, if they both infect the same host, sure. It's called interference. And sometimes when you're infected with two different viruses, the disease is very different than the disease caused by either one. So your 11-year-old is a future virologist, okay? And Amy would be happy to train them.
I would. As long as we have some money.
Catherine is a current graduate student in virology. I'm quite worried that so many people are afraid of the vaccine. What do you think? Do you have any thoughts, Amy? On this?
I think that they're worried about, I think those people are generally worried about most vaccines. I could be wrong. I think time will change some of their minds because you'll be further and further out and they'll have more and more evidence that there was nothing to be afraid of. And that the disease is much more detrimental.
Agreed. I think slowly people will turn around and the number won't be that great.
Yeah, and I think that what we will have learned is we need to not walk around saying, well, this is a new vaccine by new technology. That's not a good marketing campaign.
Thank you, Vestfield90, for your super sticker. Really appreciate it. Charlotte teaches art drawing and art history. Cool. I used to love art history in college.
That's why she was at the Calder at the moment.
Mm-hmm. Would Amy rather fight one horse-sized duck or 10 duck-sized horses?
What?
I don't know what it means.
I don't know what it means, but I'm not having a fight with anything.
Were you an undergrad at University of Louisville?
No, I was not.
Amy went to Emory.
I went to Emory, and this man is at least 20 years older than I am.
He's infatuated with you. He's
it's
infatuated.
Cute. It's great. But
Some epidemiological research shows that respiratory viruses can compete with each other in a way that means one suppresses the other. Yes, absolutely. And so they can fight to answer the 11-year-old's question. This is Eddie's favorite day of the week, TWIV Q&A. By the way, my parents got second dose of Pfizer. Me, I'm 2 weeks since vaccinated with J&J. Good. It's all good. When I get 100K subs, can you play the guitar? What do you think, Amy? Should I say yes?
No. Do not say yes.
And what happens if I play and I'm horrible, then everyone says goodbye and they leave? I would rather not that happen.
We're not playing the guitar. The guitar is for show only. It is decorative. It is a tchotchke that we can get rid of.
I used to play it. Amy, I used to play it.
Yeah. Whatever. It's a tchotchke
you didn't know
we
me.
can get rid of.
Look, you didn't know me when I played it, okay?
If you were that good, you would have kept up. So it's a tchotchke that we can get rid of.
All right.
We don't need tchotchkes.
Amy has thrown down a challenge. I'm going to play.
I haven't thrown down anything.
She thinks I sucked. She says I sucked. Last time you said the piece of protein from coronavirus that the cell presents to the immune system hasn't changed even with all the variants. Is it still true, and do we have more data on this? Oh, you're talking about the T-cell epitopes, probably. There's no more data since Alessandro Sette said that most of the epitopes are not changed, and the variants are pretty much the same as they were. So it hasn't changed. It took 600 years for Galileo to receive an apology, and yes, he was dead. What is that in referral to? Did we talk about apologizing, Amy? Oh, Hosterholm. Maybe not. I don't know. Sorry. Fully vaccinated, masked, rehab gym or no. Temp checks, hand sanitizer required, 6 feet distance, some ventilation. Okay. Well, you go to the gym, right, Amy?
I do. I go in very early in the morning or very late at night when nobody is there, basically. It's fine.
I have had COVID. It was horrible. Had both Pfizers. I work with COVID patients. I do not see an end. No, Anna, there will be an end. Absolutely. Infectious diseases don't go on forever. And we have great interventions. Ellen says, which variant of concern is most unlike the wild type? You mean, what do you mean by most unlike? How many mutations? Different? I would have to look. I don't keep it in my head. There's a great website, covariance.org. I'll put it in the chat. Let me make sure it's the right one. Do you know this site? Yeah,
it's by the lady from Bradford Slab, yeah.
And you can see what's up with the variants. Is ADE a major concern with COVID vaccinations? I only remember hearing about it as a serious problem in dengue vaccines.
That's because that's the only place it is a serious problem.
Yeah. It's a theoretical issue with others, but it's never been shown, at least in human vaccines.
It's never been shown for any other disease, despite what other people have published in the past, where they have refused to correct themselves.
You mean like Zika?
Yeah, and there's one specific group that should correct themselves, but refuses to.
I have to shout out Renzo Dalla Via. Renzo and I chat on Instagram all the time. It's really nice to see you. Buonasera.
Is he in Italy?
He's in Canada.
Oh, okay.
He's very. He's got a lot of books. He reads a lot. It's great. We have good chats. Thank you for coming here, Renzo.
Right.
Are the PEG lipid nanoparticles in the mRNA vaccine immunogenic? Could the body develop anti-vector immunity, that is, antibodies to the lipid nanoparticle, possibly compromising future booster dose efficacy?
No, they're not. I would assume no, because they have to fuse. So there has to be some kind of similarity to the composition of the membrane as there is to your cell. Otherwise, membranes don't fuse.
Not to add to conspiracy thinking, some Australia media is reporting possibility of a Wuhan lab escape. Well, I would say the Australian media is like 6 months behind the
Six
rest of
months,
the
try
world,
like 18 months.
and they're just following up. I mean, there's nothing to it. Nothing to it. Where do I find smarter friends?
If there were only a website called smarterfriends.com.
Well, you can start right here on this live stream. There are a lot of smart people here, and maybe get to know them.
For
Maybe
sure.
some of them are near you. Are there any trials of COVID vaccines for the new variants? So
Yes.
there are. Are they in process, Amy? Do you know?
Yes.
Yeah, so Moderna was the
Moderna.
first to announce it. Yeah.
Well, they also have a phase 1 already done.
You know, it's easy to do for the mRNA. It's easy to retool it right away.
Mhm.
Piece of cake. With all the press coverage on not being able to get to herd immunity now, what are your thoughts on what we need to achieve, and will we get there? Amy and I have different views on this, right?
I don't know. Why don't you say your view and I'll see if I agree with it.
So first of all, for them to say we will not get there is absurd. There's no basis for that. For SARS-CoV-2, you can calculate what you need. You need about 70 to 80% herd immunity. That's the upper end. I think that's correct, and I think we'll achieve that. And so I do not agree with the press coverage, but that's nothing new.
Yeah, I think we'll get to herd immunity. I'm not clear that I would have used those numbers because the calculation is flawed. But I I see no reason why we wouldn't get to herd immunity.
Yes, Sandy says the Moderna dose is higher. That's right. There's more RNA in it, right, than the Pfizer. That could also contribute to the side effects besides
It's
the lipid
RNA.
composition.
It's
Yes.
RNA. Well, really? You think 100 molecules out of all the RNA that's floating in your cell is really going to make that big a difference?
No, I'm saying the dose is higher. Pfizer is 30 micrograms and Moderna is 100. That could give you more side effects. That's what Sandy is suggesting.
I think that 30 micrograms and 100 micrograms of RNA, and when
Mm-hmm.
you put it in a cell, if you is minuscule concentrations of RNA compared to what is already present in the cell.
Okay. All right. So after Samantha, I said, I tested positive. How can I preserve my nasal swab for sequencing a year later if my hospital can't do sequencing now? Yeah, you have to freeze it, right, Amy?
Yeah. Okay. Sure.
Put it in the freezer. Thank you for your super chat, the Latinist. Is there any idea why there's a Moderna rash but not a Pfizer rash? I don't know the answer, but it could be the formulation, right?
I would think so.
Yeah. Is there cheese in the vaccine? No, but there should be. It would be tasty.
It would make me sick. It would not be a good idea.
Oh, thank you, Michael, for your super chat. If the comedy show Golden Girls makes a comeback, would Amy consider taking the role B. Arthur played?
I have to consult my life consultant about this. I'll get back to you.
And we have a lot of people now saying how much mold to cut off, an inch. I do much less. I want to eat more cheese. Mold is okay. Cut it off. Okay, now what about the EDTA could cause blood clots and vector vaccines? I don't quite understand this. Do you?
No, because EDTA is an anticoagulant. That's why we coat the tubes when we go bleed the mice on Friday with EDTA, so there's no clotting. So
And
how would it cause clotting?
Yeah. Is there EDTA in the vector preparation?
I don't know. I don't know, but it doesn't. It's not a. It's an anticoagulant.
Okay.
It cannot. Do we not add EDTA to our tubes because their tubes are too small to buy separator tubes.
We do.
And when we went to see Bev, she said to you, "Oh, you blah blah blah," and I said, "Oh yeah, we're adding EDTA because we
Yeah.
don't want the blood to clot. We want the serum."
For sure.
Okay, so how is it causing blood clots
It's not.
if it's an
It's
anti?
not.
Okay, so I don't understand the question.
Amy, is it safe to cook and eat an egg that was cracked in the store?
I would never buy a dozen eggs that one was cracked in the store.
Yeah, but maybe they didn't see it and they brought it home. I wouldn't either. I would throw it out.
I wouldn't eat it.
Eat your moldy cheese with salt rising bread. Clostridium perfringens of gas gangrene frame is used as leavening agent in the bread. Is that right? I know nothing about food except that it tastes good.
I'm not. It sounds correct. Yes.
Wild Bunny had some mozzarella today. Gave it to the dog because it tasted off. Why would you give it to the dog if it's off? If you don't, is that bad, Amy, to give it to the dog?
I don't know. We can ask about Gordo, but I'm sure Wendy doesn't give the dog any bad cheese.
I would equate scaremonger scientists with investment advisors talking their own book. Some of these guys sit on advisory boards of drug therapy research companies, higher profile, more board offers. That could be too. Are you okay with that, Amy?
Well, I mean, we know someone who higher profile has done wonders for their career, who's a questionable scientist. So sure. Okay. We could go with it.
All right. Amy brought up giant viruses, 10 times larger than average viruses, have metabolism genes that can first host into zombified energy factory. Are these the kind that make you sick?
No, they live in the sea.
Yeah, mostly they
live in
infect
the
amoeba
sea.
and other things. They don't make people sick. They don't. My kids are 5 and 6. The school district is forcing in-person instruction. What's the latest safety information for children that small?
I think Daniel thinks that they'll be vaccinated by December.
Yeah, but that's too late for the fall. Kids can get infected, obviously, and they can get sick, although less frequently than older people. But they're going to have to the teachers will all be immunized. They're going to have to mask. They're going to have to distance. And testing has to be part of safe school return. Super chat. Thank you, Arthur, for your super chat. Who said it is better to keep your mouth closed and let people think you're not a fool than to open it and remove all doubt?
Who do you think said that? Who do you think? Really? Really?
Who said it? You?
No, really? You never would. Like,
I think you did say it, didn't you?
of course I said it. Of course I said it.
No, there's no cheese in the vaccine. Mark Twain. Did Mark Twain say that? Really? I thought Amy said it. Well, maybe she was channeling Mark Twain.
Mark Twain said something similar.
Is it okay to delay the second dose of mRNA? Yeah, you can delay it if you'd like. Because as Amy said, the 4 weeks, 4 weeks is artificial to do the trial. Mm-hmm. In Chile, we're vaccinating people with Sinovac. It's an inactivated virus, yes. Since it's less effective than the others, would it make sense to revaccinate with those later? Someone asked this of Daniel, and he said it's not a problem if you'd like to be revaccinated. Some people have been living overseas, they got Sinovac, and then they come to the U.S. and they want to get a different vaccine. It's fine. It's not a problem.
Mm-hmm. Okay, it's 8:45. 2 more questions, and then I have to go plate myself.
We should ask Elio about the cheese issue. All right, I'll ask Elio next time. I've been cutting the mold off for years. I don't eat it. But obviously there's a little bit of mold left, and I'm fine. Look at me. I'm the epitome of health. picture of good health?
He who just made a GI doctor's appointment.
Well, I have to have my 5-year checkup,
Okay.
Does anyone complain about vaccines not being vegan, like influenza vaccine use eggs? Well, you can get flu vaccines from cell cultures now, so that's not
It's
a
not
problem.
vegan. Vegan
Cell
means it
culture?
can't have, it can't be of any animal thing. So that would have to be made in a plant.
I
I
don't
think we're
hear anyone
taking this
complaining.
idea. I think we're taking this idea a bit out of control.
The vegan idea?
Yeah.
I haven't heard it, though. How
Just
many
a matter
days
of time.
after being infected with mild COVID would a PCR test still show up positive? PCR seems to show up positive well after infectious virus is gone. Yeah, you can. You could be positive for 2 weeks or 3 weeks. You could not be infectious, though. You're only infectious for maybe 7 to 10 days after the onset of symptoms. What do you think,
Well,
Amy?
I think that there's a big problem with this question because the question is based off of the idea that load corresponds to disease, and there's no correlation whatsoever.
Yeah. Yep.
So
Are you good? Do you need to go now?
I need to go plate out my cells
All right.
and stuff.
Thanks for
So
joining us, Amy.
my pleasure. I will see you tomorrow. We're doing more mice.
Okay.
All right.
Bye-bye. Thank
And
you for
there's
joining us.
bye.
Bye.
There's something else, but I forgot. Whatever, it happens.
Bye.
All right. Bye.
Okay. Let's see. I'm a long hauler. Any chance my immune response was so bad due to viral persistence? There's not a lot of evidence for viral persistence in long haulers, which doesn't mean it's being missed. It's possible. But I think the long haulers have, out of balance immune reaction. So if you get a shot while you're out of balance, then you're going to have an unbalanced reaction. That's what I think. Now, in many long haulers, some of the symptoms seem to go away with the vaccine. So I guess yours have not. I'm sorry about that. Super chat. Oh, thanks, James, for your super chat. I really appreciate it. And no, you can't catch COVID from cheese, unless I guess if you're positive and shedding and you sneeze on it and you immediately hand it to someone else to eat, I suppose you could do that. I've never heard of such a thing. Could viruses frozen for millions of years in permafrost still be viable if released from melting permafrost? Could they be a source of future pandemics? So this is a question that was debated a number of years ago when a French group, Jean-Michel Claverie and Chantal Abergel, they took an ice core from Siberia, which was 35,000 years old. They brought it in the lab. They thawed it out. They took the water, they put it on amoeba in the lab and outgrew giant viruses. And so Claverie got in the press and said, the frozen wastes of the world could be full of viruses pathogenic for humans. I know it's not a great French accent. And so I don't know about millions of years, but possibly, but would they be pathogenic? I just don't know. But yes, as the ice caps are melting, I suppose viruses could be released, but someone would have to be around to encounter them. Otherwise, they're just going to degrade. So I don't know what we do about that. Thoughts on variant B1617? I think all the variants are should be tracked and see if they have issues with antibodies blocking them. But as we've said before, the T cells do seem to handle the variants well. So I'm not concerned about them escaping vaccines or any other variant escaping vaccines. Sorry, Amy left. We'll have to ask her next time what her favorite pizza is. Did Amy say that Pfizer also caused thrombosis? Yeah, there is. If you look at the, what's the article that I had up the other day? I could pull this up for you here. I have there's a link to an article that here we go. Anyway, there's an article that looked at the incidence of clots with the Pfizer vaccine. So there was a big brouhaha about this in the media where J&J said, oh, Pfizer vaccine caused clots. And then Paul Offit said, that's not nice of you to say that. But in fact, there were some clots, although they may have been different kinds of clots. So, but yes, there were some associated with Pfizer mRNA vaccination. If someone gets a AZ for dose 1 but supplies run out, do you think it's a good or bad idea to get Pfizer for dose 2? Well, the official line would be we don't know because it hasn't been tested. But in my opinion, it's absolutely fine because in both cases, you're getting spike protein made. So I don't think it's an issue. My 3-year-old asked, what makes virus grow? Well, virus crawls inside of a cell, and that's what makes it grow. That's a good way to tell it to a kid, right? A virus has to crawl inside. It can't just sit around and grow. It has to crawl inside of a cell and get the cell to make more of it. Yeah, the UK is doing the mixing and matching. We talked about that a long time ago. I'm a nurse practitioner. I see many long COVID or problematic disease processes. I'm worried for disease progression. Yeah, I agree with that. I think it's the tip of the iceberg, right? I think a lot of people are going to end up having long COVID and suffer for quite a long time. So yes, when we say, the pandemic will be over, what we're talking about is, the major transmission phase, but we're going to have after effects for a long time. Absolutely. And that's why NIH is putting $1.5 billion into long COVID research. We need to figure out how to treat this. Would you mind commenting on recent COVID-related deaths and severe cases in fully vaccinated? So the CDC report said there were less than 6,000 cases, and most of them were mild. So remember, not everyone is protected when they get vaccinated. First of all, not everyone responds, and the efficacy of the vaccine isn't 100%. So some people are going to be infected. In most cases, the infections will be mild because there is some immunity that reduces viral reproduction. So that's why the disease is mild. I'm not sure what is going on in cases where the disease is more severe, except that maybe they never responded to the vaccine. Dr. Conley says N95s have health risks and contests aerosols as a prime transmission. Well, not aerosols, so droplets. Aerosols are the smallest droplets that travel long distances, and that's not a source of spread. The source of spread are large droplets that fall to the ground within 3 to 5 feet. And those are stopped by masks. So masks definitely work. Hand washing helps so you don't contaminate your eyes and nose, and distancing. Surface cleaning to a lesser extent, because if you keep your hands away from your face until you wash them, surface cleaning is not an issue. If virus can last on surfaces, so traces for 3 months, but infectivity lasts no more than a week. But just wash your hands, right? I think that's fine, and I try to do that. I reckon that people who are anti-vaxxers are only able to do so because they haven't seen measles and polio. Yeah, in the old days, when I was a kid, everybody got measles. Everybody got chickenpox. Yeah, and maybe they haven't seen that. Oh, the infectious diseases are gone. So I don't know what motivates anti-vaccines. If they're genuinely afraid or misinformed, I really don't know. My niece is concerned to get the vaccine because she has the BRCA gene. Would there be any issue with the vaccine in this disease? There is no issue, as far as I'm aware, that would make more vaccine reactions in someone with the BRCA mutation. So, no, you can tell her there's no evidence that this is an issue. Spain is also mixing and matching. Good. I think it's all going to be fine, but it's good that they are checking it. Yes, open the windows of the classroom. You get better air circulation. That's good too, because not all classrooms have good air circulation. It's a good idea. What I want to play is Little Wing. I'm not sure I could get up to that. You would probably turn it off, but Little Wing is such a great guitar solo, right? Just an amazing. Efficacy of Chinese Sinovac. So we don't know, right? Which one is this? Is this the ad vaccine or the inactivated? I don't remember. Because we did review one of them. Sinovac here, yeah, that's the one which is just above 50%. And I haven't seen the data. I've seen... I want to know what this is, folks. Tell me what Sinovac is. Crowdsource this for me right now. What's Sinovac? 1, 2. Let's see if I can do it. Is Sinovac ad vector? One is inactivated. One is an ad vector. Okay, here we go. Sinopharm and Sinovac are inactivated. Thank you. So we did go over the inactivated data some time ago, but recently they said it wasn't very good. So I'm not sure what's going on. And the inactivated vaccine did very well in India. It was just out today, I think. And it's a different vaccine. It's a different manufacturing. It's a different population. So we shouldn't be surprised at that. Play the COVID blues, right? I'll make a song. Amy can wear her favorite shoes for 100K. Well, all of you will be part of it, right? Thank you, Ditching the Grind, for your super sticker. Really appreciate it. What level of change would you need in the spike for immunity to be not good enough? So here's the story. I mean, with the J&J vaccine, which is a spike vaccine, in South Africa, the neutralization titers went down tenfold, which is quite a bit. Yet that vaccine was 100% protective against death, hospitalization, and death. So more than tenfold, because the T cells are going to take care of you after the antibodies are unable to handle it. So how much more would it go down? I don't know if the spike will sustain the change. Who knows? Maybe 24. We're already past the level where the flu vaccines are changed, right? Hemagglutination inhibition titers go down less than tenfold. They change the vaccine. So obviously it's different for SARS-CoV-2. It's hard to predict. Oh, that's cute. Amy would train and be smiling ear to ear. Yeah. She's tough, though. She's tough. Here's Vinny Rack getting second dose of Pfizer. Great. By end, here's a good one. By end, do you mean it will be endemic? Yes, it will. SARS-CoV-2 will be endemic in the human population as rhinoviruses are endemic in influenza and common cold coronaviruses. Yes, it's here to stay. There'll probably be outbreaks every year in unvaccinated people. And we'll have to see if we need to be revaccinated every few years or not. If you guys believe the adenovector is the issue, then should they look at the codes to tweak them? So adenovirus vectors are known, so they've been used a lot in gene therapy trials, and they're known to bind platelets, right? And so I wonder if that's involved here, but it can't be the whole story, because what's happening is that the people who get the adenovector vaccines are making an antibody to platelet factor 4, which then triggers the clotting. So I don't know if that has anything to do with it binding to the platelets. I'm looking into that. I want to make a video about this to try and explain it. Oh, you're a shoe artist. How cool. Amy would love that. Lancet published a paper indicating it's primarily spread by aerosols. This is nonsense. Aerosols are going 100 feet or more. That's just not, that's measles, not SARS-CoV-2. SARS-CoV-2 is droplets, intermediate and small and large droplets. Amy said her favorite ice cream was cinnamon. Yeah. Is it okay for the Indy 500 to go ahead with 135,000 fans? Oh boy, I'm not touching that one. If they're all vaccinated, that's crazy to put them all together. You're going to have a big outbreak if they're not vaccinated, right? Are the blood clots caused by J&J more concerning than AZ? No, they're similar serious brain clots. You don't want to have that happen to you at all. That's my thought on the situation in India. I think there are, well, remember a long time we said, why is there so little COVID in India? Now we see it's taking hold because there are a lot of non-immune people. And so they're just having a big outbreak. There's a large fraction of the population that's not immune, and the virus is there and it's spreading. It's nothing magical. And it happens to be a couple of variants that are there associated with it. So people are blaming the variants, but it's not. It would have happened the same anyway. I'm surprised you said on Twiv animals don't get polio. I've been working on polio for 40 years. That's true. I saw a nature show claimed a baby primate died in the wild from polio. No, that's wrong. I mean, Jane Goodall thought her chimps had polio, and she actually vaccinated one of her colonies, but it's not true. Chimps do not get polio in the wild. Nope. Can I do an episode on COVID in India? Oh, yeah, I heard there's not enough oxygen. This is tragic. That's a real problem. But people are trying to help, I can tell you that. I don't understand why no one seems to link the ability of coronas to form multinucleated cells or syncytia and their affinity for recombination. Aren't these complementary? Not necessarily. So to get recombination, you just need one cell, right, infected with two different viruses. So I know what you're thinking is, say you have two infected cells and they fuse, then the genomes are all going to be in the same cell. I got that. But you don't have to do that. You can just have two viruses infect one cell. And in fact, on Tweevo today, which we live streamed, we did a report in virological.org, which said that there are a lot of recombinants of B1117 circulating in the UK. So recombination is happening for sure. People are working on getting India help. I know this behind the scenes. It's happening. Don't worry about it. Well, you should worry, but people are not just sitting around for sure. Using Galileo as an example, bad idea. He argued well, but he had not proven it. Dr. Goldman, but scientists know, lecture on YouTube, historian of science, vaccines have good proof. What about a Twix Discord server? Yeah, I think that's a great idea. Yeah. That would be a chat thing, right? I don't do Discord, but I'd participate, sure. So tell me what you think of this. I did an AMA a couple of weeks ago on Reddit, and they want me to do one every week after a TWIV to go on and take questions based on the TWIV. What do you think? Should I do that? I don't mind doing it. I'll say yes to anything that can teach people. Is it feasible to create a vaccine before there's a pandemic? Yes. Yes, it takes money. That's all. So you go in the wild, you sample coronavirus, SARS-like coronaviruses in bats, and you sequence their spikes and look for conserved regions in the stem of the spike and see if they induce broadly neutralizing antibodies. So you do this in animals, and you have to have good wildlife sampling to do that. But yeah, it couldn't be done, but there was no incentive. You can have antivirals that would be pan-coronaviral too. I think we need to do that before the next coronavirus pandemic, don't you? Yes. I might play with 100,000 on YouTube. We're at I think today we're approaching 89,000. So it'll be a while, probably in the fall, I would guess. It's 50%. We'll get to 90 in the U.S., I guess you mean. Yeah, I think we'll get to 90%. I have a feeling that the vaccine reluctant people are not going to be more than 10%. Don't fear the weeper. That's a good song. That was Blue Oyster Cult, right? They were from Oyster Bay, Long Island, if I remember. Yep, a local band that made it big. What do you think about the vaccine boosters that are being developed as a possible yearly shot? I think it's great that we're doing that, and you could view it as a new, like a flu vaccine changes every couple of years. Is it going to be the similar thing? We're going to look at the circulating SARS-CoV-2 variants and say, is it more than 10-fold neutralization drop? Maybe 20-fold. I don't know what the number is going to be. And then have a new vaccine. I think it's a good idea. Now, we do have to see how long the current immunity is effective, right? Because you don't want to make a booster if you don't need it. So very interesting times coming. Stay tuned. We'll talk about it here. This is a good question. We talked about this a little on Tweevo today. Jesse Bloom is trying to see the mutational space of the virus, but actually he's focused on the spike and he's focused on the receptor binding domain. And in terms, for example, of just interaction with the receptor ACE2, a lot of changes, and I forgot the number, aren't tolerated. So not every residue can change, only a fixed number, and that number evades me at this moment. But I don't think we've seen all of them yet. There are likely to be more. And then, combinations can have different effects as well. But, it's not an exhaustive study, I think, because just receptor binding is not the only, it's not the only determinant. What are your thoughts on the April 20th Nature Medicine article that observes higher B cells in asymptomatics, but more T cells in moderate and severe? So that's interesting, and that would make sense. Yes, we'll take a look and cover that. Let me make a note of that here. B cell, T cell paper. Nature 20th April Nature Medicine. Okay, upcoming TWIV. You got it. I saw someone say, "Oh, well, I'm sorry, Patricia. Amy left. I want her to answer that." Is it true MERS basically went stealth? No, it's still. I mean, cases have declined. So what happens with MERS, of course, is a camel is transmitting it to people. So camels are all infected at birth. They get a respiratory infection, and then people who are in contact with them, which is common in the Arabian Peninsula countries, they get infected. So there usually were a few cases a month, and it's really gone down quite a bit. And it may be because people are staying away from camels. I doubt it. I don't know what's causing that. It's still there. It's in the camels, so I don't think it's gone away. Can dehydration cause blood clots in the vaccinated? I don't know the answer to that, but in general, it's not good to be dehydrated, and one of the reasons is for clotting. Yes, so okay. I'm just passing through quickly here to cheese. I hear conflicting stories about whether or not Pfizer or Moderna. So as I said earlier, there is an article which I was not able to find because I don't want to take the time to do it. There is an article comparing them, but you can go to the documents that were submitted to the FDA. They're online, they're public, and you can see all the side effects. I'll do that for Friday's TWIV, okay? Clots for Friday's TWIV. Okie dokie. Boy, would I love to have an assistant who could help me do research for TWIVs, right? The problem is volunteer doesn't work because volunteers eventually stop volunteering because they're not paid. So maybe I should pay someone to do that. Anyway, Liz, at least one of the others had clots associated with it. Maybe not the same kind, not the cerebral clots, though. I have to check that. Good point. My second dose was injected rather high in the arm near the shoulder. Some blood and pain almost immediately. Is it possible the deltoid was missed? Will this affect efficacy? You want to get it into the muscle, right? It needs to go deeply. Near the shoulder, it could have missed it, yeah. Second dose, did you get a more severe reaction? You could sort of get an idea. Maybe at some point you want to check your antibodies and just get another dose. Rhinovirus. What's the silliest but coolest lab accessory you bought just because you wanted it but not so much needed it? Years ago, we bought a microtome. You know what that is? It's a big machine for slicing tissues and looking at them under the microscope. Never used it. It was like $5,000. Should never have bought it. Should just have used common facilities at Columbia. What about social distancing training to a new office worker? HR thinks it's okay to use the same computer. Well, if you're masked, and maybe you're also vaccinated, you can. It just should wash your hands after using it. Don't touch your face afterwards. I think that's fine. That's an old saying. Yeah, I didn't think she said it. And I don't claim to say anything. Somebody from BioNTech says the third shot will be needed as immunity wanes. I don't think there's any evidence that immunity wanes. First of all, immunity always wanes to a low level. That's what memory is for. You have low levels of memory cells, and when you get infected within a couple of days, they immediately make antibodies and T cells, okay? So to say it wanes is kind of not right. Now, if the memory cells went away, that'd be a different story. And I don't think that Dr. Turici knows whether they go away or not because it hasn't been far enough out. Now we're just getting to the Amy good night. And vegans. Can a vaccinated person become an asymptomatic spreader? I don't think so. I think it will reduce the amount of shedding sufficiently that you will not transmit. Otherwise vaccines wouldn't give herd immunity. So the rhino question is, why is rhinovirus coming back? If you go look at the BioFire website, you can see rhinovirus infections are up, compared to flu. Why is that? And so Amy is going to answer it because she works on rhinovirus. And so I don't want to answer it here. What happens after the vaccine is in a muscle cell? Well, it gets picked up mostly by antigen-presenting cells, which are dendritic cells and macrophages. The protein is made, and then those cells present it. They go to the lymph nodes in your armpit, and present the peptides derived from the spike protein that they've made in response to the vaccine. They present those to T cells in the lymph node. T cells will recognize them as foreign and become activated and initiate immune responses, which involve making antibodies and cytotoxic T lymphocytes. Thank you, John Snyder, for your super chat. Quick Amy left. Break out the guitar. No, we have to get to 100,000 subs. And then, I agree. Someone said I should play even if Amy says I shouldn't. Yeah, of course. Of course. Even if produced in plants, hasn't Medicago used animals for toxicity studies of their VLPs? Yes, of course, they have to. And how would vegans feel? I don't know. Yeah, but they make virus-like particle vaccines. They certainly had a flu virus-like particle vaccine. And yes, it have to be a plant cell line to be vegan. So you could make the vaccines in plant cells, yes. But if you test them in animals, is that a problem? You've said that vaccines should still prevent serious illness against variants because T-cell epitopes have changed. is this killer T cells. This is killer T cells, right? Because they are recognizing epitopes presented on the surface of infected cells by MHC molecules. Yeah, they're not changed, and they're going to save us. T cells are going to save us. Even bacteria can survive for millions of years. I know you're giving this to someone else, but it's a good thing to share. I like that. Dr. Griffiths said on Twitter, 40% of our long COVID feel improved. Yes, he also said it on Twiv. 30 to 40%, he said. I think, here are the three theories. Psychosomatic, which I don't think is correct. Elimination of some virus that's still been replicating that we never detected. I think highly unlikely. My opinion is that it puts the immune response back into sync. It's out of sync for whatever reason, and this does it. I don't know how. That's what I think. But as you know, Daniel Griffin also said, if you have pulmonary damage and you're a long hauler, the vaccine will not help you because it cannot reverse that. Yes, meteorites from space could cause viruses like when the block. Yeah, okay. Yeah, now we're just getting to the ice melting issues. You haven't read any article. What kind of articles you want? You want science articles? Look up Jean-Michel Claverie. We covered it on an old episode of TWIV. What degree does someone need to work in virus research, vaccine development? Well, it depends. I mean, you can have a bachelor's of science degree and get a job as a laboratory technician and work in research, virus research or vaccine development. Sure. Now, the higher you get your degree, the more pay you're going to get, the more responsibilities and so forth. So, but I started as a technician after I graduated college and I loved it. I decided to get a PhD. It's a good place to start. Why don't we add 3 dogs before questions so that Vincent can filter them? You could put something in front of my questions for me, right? And that would help. Because right now I have them scrolling down on the right side. And that's why they're little pauses. I'm sorry, I don't like pauses, but there you go. What about the prions? Well, one of my last lectures in the course. Check it out. Lecture 24, Unusual Infectious Agents. I thought CDC recently said aerosol transmission was a factor. It's limited, very limited in certain situations. You know, as I say, they may be referring to droplets. And as Doug says, airborne means droplets, particles. Aerosol is vague. Some people use aerosol to mean the tiniest droplets that travel the farthest. Okay, I don't use it that way. I think aerosol is, I don't use it at all. I use droplets for everything. There are 3 different sizes of droplets, big, medium, and really small. And that's how I look at it. Droplets involve aerosol. I'm not going to use aerosol. The tiniest droplets, less than 1% of the cases of transmission, I would say. And I think that's what the CDC said. Why don't Western countries use the Russian and Chinese vaccines? The FDA will not license them, at least without some of the trial being done here in the U.S. Well, I'm just kidding about that. I know you wouldn't. You know you wouldn't do that. Now I'm getting the answers. You guys are slow on the answers, but I'm slow. Actually, the stream is very delayed. Is there any issue with getting a 3rd shot? I don't think so. I think it's fine. If supplies are not constrained, yeah, you can get a 3rd shot. It's not a problem. DR, thank you for your super chat. Amy's wrong. Here's the one I was looking for. If playing guitar brings you pleasure, play, regardless of how skilled you are. I think I haven't played in a while because I've been interested in other things like teaching virology, right? So I have a limited amount of time. And like today, I spent the whole day doing this. I did a live stream with Nels. I did editing of TWIV and twin, and some other things, emails with my course, and that's the whole day. If droplets are the mode and they fall down, why do we need good ventilation? Only aerosols stay suspended. Well, aerosols, the tiniest droplets, are not the mode of transmission. And the idea with ventilation is to get those droplets out, I suppose. My theory of the variants and localized spread is that they are the virus's adaptation to the population. The Indian variant will not have the same impact in the U.S. So I think what it is you're right in a way. The variants have better fitness compared to what was there before. So they displaced the previous virus. But what confers the better fitness isn't clear. One possibility would be droplet transmission, but there are other possibilities as well. So you can't assume it was droplets. And I think to a certain extent, yes, one country's variant won't work in another country. But, the UK variant has shown, the B1117 has come to the U.S. and many other countries, and it has displaced other viruses there. So I agree in part with what you say. In some populations, the variants are specific and won't go elsewhere, yeah. Are adenovectored vaccines a one-shot deal? Once your body recognizes that vector, will it still be effective for a second? It depends on the vector, actually. For Chadox, the chimp adeno, it looks like the first shot doesn't give you much vector immunity that interferes with the second, which to me was a surprise. Now, the Sputnik Russian vaccine used 2 different adenoviruses to get around this problem. But I don't know if it's as big an issue as we thought it was, at least for some of the vectors. It depends. There's Ad5, there's Ad21 for humans, or 26, I forgot. And then there's the chimp adenovirus. Those are the main ones. Why did Japan ban the MMR vaccine? I don't know. What are the prerequisites for my virology course? Well, if you want to take it on YouTube, there are none, right? But if you want to take it at Columbia, some college biology is a prereq, and I have to tell you, not everybody. They don't enforce prereqs. They don't enforce them. So Eric, not every prime boost is an issue. As I said, the Chadox doesn't seem to be an issue. It's surprising. Hello, Tom. Welcome. I missed you. All right, good. AstraZeneca, very good. Do you think animals on farms have to be slaughtered? No, they're going to be vaccinating them. If you heard the San Diego Zoo lady on TWIV last week, she said there's a vaccine for minks and for great apes. And so presumably whatever mammal you're raising, you can have a vaccine for it. So that'll be taken care of. That is the smart way to do it. Does the mRNA spike have a transmembrane? Yeah, it's got a transmembrane domain. It's not secreted. And Patricia, we already did that. We discussed this earlier. I think the double, triple spike change variant is not concerning. As you heard, the India inactivated vaccine has done pretty well. It's good. Yeah. Adenoplatelet binding and antibody binding based platelet activation are probably synergizing. Maybe. Yeah, it could be. We don't know. Yeah, this is good. Are there any theories as to why adeno infections don't cause cerebral sinus vein thrombosis? Maybe they do, and we just don't notice it. But I see no theories. That's something I want to look into. Got to look into that. Too many things to do, not enough time. Isn't that what they say? Why did I choose polio? Well, I chose a postdoc. I wanted to work with David Baltimore, and he He had two different groups, retrovirus group and a polio group. He told me he wanted me to work on polio. It was fine with me. He's an RNA virus. My philosophy is I pick a mentor and then I listen to them because I'm picking them because I trust their judgment. And so that's why what you do as a postdoc, in the old days, you did as your first job. So that's what I did. Didn't regret it. I love it. Okay. We can discuss Discord with the Reddit group for sure. You bet. I'm upbeat. Yeah. Someone's answering the question. I think said his PhD prof picked it. Not my PhD prof, my postdoc picked it for me. It doesn't always happen anymore. But I'm fine with it. I did all right. Thank you, Tom, for your super chat. Much appreciated. Trying to get the studio moving forward. Don't forget to buy the textbook. You don't have to buy the textbook. You can. I don't ask you to buy anything by coming here, but if you'd like to, you're welcome to. It's a good book. A little off topic for A&V. How does it get rescued from its integration site? All right. So if AAV has a rep gene in it, it will integrate and it stays there. It doesn't get rescued. But enough viruses will not integrate. So if you want to make a vector, you take the rep gene out. You just add the adenovirus helper functions on plasmids, and that's how you make virus particles. So there's no rescue from the genome. We should decide what kind of thing you should put in front of your questions to make sure I see them. And the problem with that, of course, is that people who are new to the live stream won't know that, right? So you'll have to inform them, I suppose. Yeah, I saw John Campbell had a video on this. I don't really look at his videos. I prefer to get my own information. When you have your studio, can we have a celebration? Yeah, Amy wants to have a party. I said, why do I have to have a party? But she said she would organize it. And I have to have the big shots like Ian Lipkin and David Ho, right? And Jonathan Weiner. But you can come, yeah. We'll publicize it, and you can come and check it out and take some videos there, for sure. Yeah, you can come. I want people to visit. Just have to contact me ahead of time. And so I make the prereqs for my course. So you just have to ask me. It's just some course in biology. And you know what? They don't check. You can register, and people will often say, I don't have this. Can I take it? And I say, look at a lecture on YouTube and tell me if you understand it. And if they do, they can check. They can take it. So I am very lax about prerequisites. So you could be a virologist still. Well, I don't know where you are in your education or career, but yeah, why not? Just get Amy to train you. How risky would it be to go to a concert mid-July in LA, fully vaccinated? Well, it depends on how many other people are vaccinated, whether they require it, and whether people are going to be masked and so forth. I personally wouldn't go because even though I'm vaccinated, I don't want to have so many people around me. And this is a good way to, you can sneak into my class. You could actually, at Columbia, there are no guards that keep people out of buildings. So if you find out what room it is, you can come in. Yeah, next spring I'll teach it in person, for sure. What % of labs are funded by Bill Gates? Vanishingly nothing. We asked him for some money to help make a new polio vaccine. He said, no, we are only spending money on COVID. All right, so on that note, here's the last thing we can put up here from Anthony. Twiv long and prosper. Oh, that's great, isn't it? Unfortunately, it's stealing from Star Trek, but thank you. Twiv long and prosper, everyone. I hope you have a good evening. Thanks for coming. I really appreciate it. There have been 460 of you tonight. Amazing. Save your questions from next time. And the chat opens up a half hour before the live stream, and I'm usually here checking out the chat, and I'm talking to people. So if you want to get in your questions early, you can come there. Otherwise, have a great week, and I do appreciate your coming. Be safe, okay? Bye-bye.